In this video, we're going to start about mitosis and meiosis, the basic mechanism of sexual reproduction, and about the meiosis as well. So you have to know that the biology is the only science for multiplication and division. They have the same meaning. Mitosis, as you know, it occurs. The somatic cells, diploid cells, do N, where they reproduce and they give new daughter cells. They are identical to the mother cells. They have the same genetic information and it's conserved. Why would the body cells need to reproduce? We have two goals: to increase the number of cells and allow the growth of the individual, like the case of the zygote, and to renew the body tissues and replace the old or dead or the injured one. You have to know that the mitosis may be divided into interphase and mitosis, the cell cycle. So interphase, we have G1, gap1, as the synthesis phase where duplication of DNA or replication of the genetic information occurred. And we have G2, gap2, which is preparation for the mitosis. And the mitosis is divided into four main phases: prophase, metaphase, anaphase, and telophase, which is met. So you have to know that the gap1 as G1 as DNA synthesis or replication of DNA takes place during the S phase, and gap2 is preparation for mitosis. And you have to know what's happening during prophase. Prophase, you have to know that the nuclear membrane disintegrates, disappear. Lymphatic spindle fibers, they are formed. And you have to know that the centrosomes, they change into answers. The chromatin, they condense, forming chromosomes. They are easily distinguished. Each is made up of two chromatids each. This is prophase. Then we're going to move to metaphase. Metaphase, the hint or the special about metaphase that the chromosomes, they line at the middle of the cell, forming what's known by equatorial plate. That's why we add colchicine to stop the cell at the metaphase, because the chromosome, they are well distinguished and they are distinct, and each chromosome is made up of two chromatids each, if you remember in the karyotype. So now let's move to the anaphase. Anaphase, we have something special also, which is polar ascension, where the sister chromatids, they move out the spindle fiber, they pull the sister chromatids apart. Each sister chromatid moves to the opposite pole of the cell, forming what's known by polar ascension. Here we have two groups of chromosomes, each made of one chromatid each. So spindle fibers, they pull the sister chromatids of each chromosome apart. Each chromatid moves to the opposite pole of the cell. This is what's called by polar ascension. Late anaphase, we have two identical groups, each made of one chromatid of the opposite pole of the cell. At the end of mitosis, we have telophase and cytokinesis. Telophase and cytokinesis, the cell is going to cleave. So we have something called cleavage furrow. Cleavage furrow, the cell is going to cleave and divide into two. And cytokinesis is the definition of the cell into two cells. So as I said, the nuclear membrane will reform around each set. Spindle fiber, they disintegrate, and after they go back to the centrosome state, cell membrane constrict and forming what's known by the cleavage for the two identical daughter cells. You have to know that the process when the cytoplasm constrict, start to constrict, dividing the cell into two daughter cells is referred to as cytokinesis. You have to know that the cells have, at any stage of mitosis, they have two only chromosomes. The two daughter cells obtained, they are identical to each other, and also they are identical to the mother cell. All of them, they are diploid, and they have the same genetic information. So as a revision, we have prophase, metaphase, anaphase, telophase, and these are the phases of mitosis. We have, for example, this application showing the variation of the quantity of DNA as a function of time. So at the beginning, the quantity of DNA was at time 0, time 1, it was 4 AU. It increased to about 8 AU at 4th hour, and it remains constant till the 7th hour, where it divides into 4 and remains constant till the 10th. They said represent the data in the form of a graph. You have to draw a graph. Graph should be drawn using a pencil, should be given the title, should be given a scale, and you should erase all the unused values or numbers. For example, we use the value 1, 2, you have to erase them. Analysis of the graph, duration of the cell cycle. So at time 0, the quantity of DNA was 4 AU. As the time passes from 0 to the first hour, the quantity of DNA remains constant at 4. and then the time passes from 1 to 4, the quantity of DNA increases from 4 AU and remains constant. You have to mention these, because if you mention these, you are describing or analyzing the graph in a good way. You don't have to say, for example, as the time increases from 0 to, let's say, to 4, the quantity of DNA increases from 4 to 8. No, you have to mention that during this value from 0 to 1, it remains constant, and then from 1 to 4, it increases from 4 to 8 and remains constant till the 7th hour, the early 7th, and then it divides into the 4 AU. Then at 7th hour, the quantity of DNA drops from 8 to 4, is divided to half, and remains constant at the 4 AU till the 10th hour. Life cycle start at 0 and finish at 7 hour. So 0 to 7. This is the cell cycle, the duration of the cell cycle. All of you know that the duration of the cell cycle includes mitosis plus interphase. So part 4, they're saying that knowing that the mitosis needs 1 hour. What is the duration of the cell cycle? You have to know that duration of the cell cycle is duration of the interphase plus duration of mitosis. So 7 hours represent the duration of interphase plus 1 hour. So duration of interphase is 7 minus 1, so it's 6. Second hour it was the interphase. Early it was the anaphase, and late it was the telophase. And for 7, they said what happens during the S phase, or what happens during the interphase at the level of DNA. You have to know that during the S phase of the interphase, the DNA is replicated. Where it's duplicated, becomes double. Duplicated or replicated, the same. Note as in plant cells, there is no asters, there is no constriction, because we have a cellulose wall that is very rigid, very strong. Now let's move to the gametes, specialized cells with 23 chromosomes. So we have to talk about meiosis. Mitosis, we said, it occurs in the somatic cells, diploid cells, whereas meiosis is the process by which the mother cell of gametes, diploid, 2N, they gave birth to the gametes, haploid, 1N. Otherwise, we're going to be, every individual is going to be 4N and not 2N, because if we have, for example, 2N gamete and 2N gamete, they mix together, we have 4N, so this is impossible. So before meiosis begins, the mother cell of gametes go through interphase and preparation. Meiosis includes 2 divisions, meiosis 1 and meiosis 2. Meiosis 1, as you know, is referred to as the reductional division, and we see why it's called the reductional, and meiosis 2 is called the equational, and also we'll see why it's called the equational. Meiosis 1, let's say meiosis 1, prophase 1. Meiosis 1, prophase 1. You have to know that the nuclear membrane disintegrates. Let's compare those to the mitosis. Nuclear membrane disintegrates, spindle fibers start to form, centrosome change into aster, chromatin condense, so that in the identical chromosome they are distinguished and each made of 2 chromatids. These 1, 2, 3, and 4 steps or 4 events, they are similar and identical to the mitosis. The difference here is we have synapses, we have crossing over. You have homologous chromosomes, they pair up, they form each one over the other. So this is what is known by the tetrads. This increases the chance of the crossing over between the non-sister chromatids of the paired homologous chromosomes. Metaphase, it's identical, but here, not chromosome, they line up in the middle of the cell. They have tetrads, they line at the equator of the cell, forming what's known by equatorial plate. Here, the anaphase, not spindle fiber pull the sister chromosome. Spindle fiber pull the homologous chromosome of each tetrad apart. Each chromosome will move to the opposite pole of the cell, forming what's known by polar ascension. You have 2 groups at the end of this stage, which is anaphase 1, 2 groups of chromosomes. Each group is made up of 2 chromatids at the opposite pole of the cell. Telophase, also we have cleavage furrow, where each daughter cells are obtained, where each carries 1 copy of each chromosome, and it's composed of 2 chromatids each. Obtained daughter cells, they are haploid. Number of chromosomes is reduced. That's why it's referred to as the reduction definition, because the amount of chromosomes or the number of chromosomes is reduced into half, so it's called reduction division. Here, the daughter cells, they are different from the mother cell, and they have differences. Telophase, the chromosome, as I said, they form two groups at the opposite pole. Spindle fiber disintegrate, and the asterix goes back to the centrosome, nuclear membrane, reform. You have to note here, kindly note that there is no interface between meiosis 1 and meiosis 2. Meiosis, we have only one interphase. Before meiosis 1 interphase, meaning that G1 as G2, and then we have meiosis 1, and immediately after it, we have meiosis 2. So there is no interface between meiosis 1 and meiosis 2. Okay? Prophase 2, here we have, in prophase 2, we have two daughter cells here. The work is two daughter cells, magnetic spindle fibers start to form, centrosome change into aster, nuclear membrane disintegrates. Metaphase 2, here we have the chromosomes, they line up at the equator of the cell. There is no tetrads here. The chromosomes, they line up at the equator of the cell, forming what's known by equatorial plate. Okay, now here the sister chromatins, the spindle fiber will pull the sister chromatins. If you look here, the events they are very similar to that of the mitosis during meiosis 2. First, the chromosomes they line up at the middle of the cell, forming equatorial plate. Here, the sister chromatins are pulled by the spindle fiber to move to the opposite pole, forming what's known by polar ascension. And the late anaphase will have two groups, each made up of one chromatid. Okay, so it's very similar to that of mitosis. So meiosis 2 is very similar in its events to that of mitosis. 8 spindle fibers, they pull the sister chromatids of each chromosome apart. Each chromatid moves to the opposite pole of the cell, forming what's known by polar ascension. By late anaphase, the result will be two groups of chromosomes, each made of one chromatid at the opposite pole of the cell. Here we have telophase 2 and cytokinesis. We have each cell will divide into two, so we have four daughter cells, the final result. So the chromosome form two groups at the opposite pole of the cell. These chromosomes will decondense back into the chromatin form. Nuclear membranes start to reform around each set of chromosomes. The spindle fiber disintegrates. Okay, so here you have to know the different phases of mitosis and meiosis. You have to study the events. For example, maybe I'm going to show the figure like this. We have two daughter cells at the end of this process. What do we call this? You have to know that it's mitosis. Why? If we have four daughter cells at the end of this cell division, so it's called meiosis. The mitosis, it occurs in somatic cells. This table is showing the differences between mitosis and meiosis. Mitosis occurs in somatic cells, while that of meiosis occurs in gonads. Aim for mitosis is for the growth and for the renewal and the replacement of dead cells or injured cells, whereas the aim of meiosis is production of the gametes, which are haploid gametes, sperm and ovum. Okay, the 1N. One division mitosis producing two daughter cells, whereas meiosis it's two division producing four gametes. Mother cell and daughter cells, they are identical. They have the same number of chromosomes per cell. Why? The number of chromosomes is reduced into half, going from mother cell, which is diploid, to the gametes, which are haploid 1N. Mitosis, it occurs to somatic cells in all the body organs, like the skin cell, muscle cell, brain cells, and others. All the cells in the body except for the gametes. So while meiosis, it occurs to the gametes and the gonads. Mitosis, it's made up of one division, as we said, preceded by interphase. So we have interphase, then we have mitosis. Here, meiosis is made up of two divisions. Meiosis 1, reductional division, meiosis 2, equational division, and both of them, they are preceded by interphase. So there is no interphase between meiosis 1 and meiosis 2. You have to be careful about this. There is no pairing of homologous chromosomes during mitosis, while during meiosis, we have a pairing of homologous chromosomes. We have crossing, increases the chances of crossing over and the formation of tetrads. Daughter cells obtain the R2N, one chromatid per chromosome, while after meiosis 1, daughter cells 1N, two chromatid, after meiosis 2, 1N, one chromatid. Kindly note that meiosis 2 is very or greatly similar in its event to that of mitosis. So please, All of you, you have to study the events of mitosis and meiosis, because in the coming lessons, in the coming activities in spermatogenesis or genesis, you have to know these. You have to know what happens during mitosis and what happens during meiosis, and the different events during these different phases. Okay.